The structure of DNA. What do we know about the structure of DNA? The rails are antiparallel. We can see that we have the 5' end and the 3' end on this end. The 5', 3' business refers to the ribose. The ribose has the 1', 2', 3', 4', and 5'. The 5' carbon on the ribose is the little carbon that sticks out of the ring. That's the 5' end. The 3' end is the third one over. And on one side, they're 5', 3', and on the other side, they're turned over. So the bases, what do we know about the bases? Okay, we got thymine, adenine, guanine, and cytosine. Take adenine. Adenine, the molecule of adenine, has no symmetry whatsoever. It's flat, but that doesn't mean there's anything symmetrical about it. Adenine is always drawn with in this format. I think it could have been drawn the other way around, but it wasn't, so that's fine. It's sort of a definition for us. The adenine and the thymine are always lined up this way. They're never lined up this way, which is to say that if the double helix was indeed a spiral staircase, you would always have the same adenine, thymine facing the same way. These molecules are not radially symmetrical. You turn it over, you don't have the same thing. They're always lined up the same. So you have adenine, thymine, adenine, guanine, cytosine, guanine. They're always lined up the same, which tells us right now that DNA is not the same top to bottom. Just that one point tells us that. What else do we know about DNA? DNA has the codon and the anticodon, or the sense and the antisense, or sometimes it's the codon and the template. The two strands are not the same, or they're not used the same. They're not used the same, which again implies an asymmetry to the molecule. The RNA is transcribed against the anticodon, which gives the RNA the codon. So you have the codon and the anticodon in the DNA. If the RNA is transcribed against the anticodon, then the RNA picks up the codon, which is kind of the idea of it. The RNA, they say it's transcribed 5' to 3'. Well, 5' to 3' doesn't tell us very much because there's a 5' to 3' on both strands. The RNA transcript, of course, goes out into the cytoplasm, encounters a ribosome, and turns into a protein. Very much part of the magic of biochemistry is the question of where does the RNA start and where does the RNA end? There are proteins which promoters and repressors, and they have all kinds of different names, and there's big families of them, which spend all of their time in the major groove of the RNA. And if we look at this model, we can see that a molecule which sits in the major groove, if you turn it over, it'll still fit in the major groove. But the major groove is not the same. One side of it, in the major groove, you have a 5' and you have a 3', 5', 3', and then 3'. You turn it around and you don't have the same thing. And furthermore, the bases themselves, they must kind of stick out into the major groove. The promoters and the repressors and all the things that sense what's going on in the DNA, they, for the most part, do not unravel the DNA. And they know where they are based upon kind of feeling the edges of the bases. There are people that are studying that, but the final conclusion is that the DNA looks the same if you turn it over, but it's very much not the same.